QIAGEN ingenuity pathway analysis. Our partners on this program is Scott Magan. Is it Magan? Did I pronounce it? Magan? Scott Magan and Kristen O'Malley will be presenting this training workshop on the ingenuity pathway analysis software that the GSI purchases with our support from the Dean, Karen Antman, for funding the GSI. We purchase a big site license and then ask labs that want to use it to contribute to it. So this training session is part of this ongoing program, and we hope to continue supporting this since many of you find it useful. Before I hand it off to Kristen to do the training session, just some GSI business announcements to any of you live streaming. Please note that we have a March 17th deadline for the submission of applications for the Bioinformatics Master's Students Summer Internship Program. So if you're a lab that wants to recruit a summer student from the Bioinformatics Master's Program to work on any genomics for the summer, please submit those applications by March 17th. And April 7th is our second deadline for the applications for the pilot grant workshop for either applying cut and run or split-seq kits for advancing those aspects of the research or other genomics research programs. We have 2 seminars, GSI seminars planned in April, so look out for those. One April 7th, anyway, check out the GSI website. We'll be sending out those notices of 2 seminars in April. And then Rachel O'Neill's seminar in February was postponed because of a snowstorm that affected Connecticut way more than affected us. We saw practically no snow, but at Connecticut they did get quite a bit of snow, so that's why she postponed. We're hoping to reschedule that either in late March or May, so stay tuned for that. All right. So without much further ado, I'll hand it off to Scott and Kristen to tell us about QIAGEN IPA.
Great, thank you. So as you guys know, we do this pretty frequently. We try to do it a couple times a year if possible. We have a lot of canned trainings. We have different topics we cover online if you've ever visited our website. Whenever possible, though, we do like to get concepts, ideas, questions, data sources, things you guys have in mind. So we use that to shape and to tailor trainings. If ever you run into a problem with the software, you have routine technical support, but we're also available. So Kristen's part of a team of field application scientists, and they literally spend almost all of their time rolling up their sleeves and looking at what different data scientists and biologists are using the software for. And the feedback that you give us is useful in helping to shape the product. So part of our job is to remain vigilant for what people want to see. One of the things we also do with Ingenuity Pathway, so for those of you who haven't seen the talk from us before, we always emphasize the fact that it is based on an ontology of findings derived from manually curated literature. So anytime there's a cache of literature that's relevant to your field that is not represented in the software, you can always suggest it. Email us, say, by the way, this is really important to telomeres or something, and we will take it under advisement that the underlying database is updated every week. So these are things that can be eventually incorporated and will turn up. I know a lot of academics tend to look for their own stuff, so by all means let us know. You're welcome to follow along with the training, but that's not any kind of requirement since we're recording it. And like I said, we're happy to follow up with you. Both she and I are local to Boston, and we have a number of other colleagues that visit the area periodically as well. So anyways, looking forward to hearing your feedback. Feel free to interrupt her with questions. She loves it.
Love it.
Definitely. We're a little bit of an abbreviated timetable today, right? Basically,
yeah.
2 sessions. I'll let you speak to how you want to divide that up.
Yeah, so what we're going to do today is I kind of divided things up into 2 sessions. So the beginning part of this talk will be some of the basic stuff. So we will zip through data upload, how to get your data into IPA, and then we'll go through things like canonical pathways, and touch on upstream regulators, and then I'll show you guys how to take multiple comparisons and put them all together into the same view so you can look at multiple core analyses together. And then if we have time during that, I'll also show you how to use IPA to construct networks from scratch. So this is a really cool opportunity to maybe try out some in silico testing or figure out what drugs are targeting a specific target that you're interested in. So that'll be the first half of the time spent together today. Second half, we're going to focus more on some advanced topics. So maybe we'll dig a little bit more into upstream regulators, and then we'll talk about some of the really cool features in IPA where you can take your own data and patterns that you see in your data and match it to publicly available data sets that have been uploaded into IPA. So things like analysis match, if you've heard about that before, pattern search, activity plot, we'll go through that. And then we'll also touch on this other cool feature that's kind of plugged into IPA called Land Explorer. And that's where we can go and look at this publicly available data kind of in a more granular way. So we'll, hunt for analyses that match our own, and then we'll go and look at the volcano plots of all the gene expression data coming out of these publicly available studies. So we are a little bit on an abbreviated time frame, but I definitely don't want that to deter you guys from asking questions. That's what I'm here for, is to answer your questions. And of course, that doesn't end today. If you guys have any questions as you're using IPA, feel free to let me know. I think I have my email is in numerous places throughout the presentation here. So don't be a stranger and reach out whenever you have questions. Additionally, I'll send you guys these slides after the workshop here, and I have included some step-by-step guides on some of the more kind of nitty-gritty of like the data upload, because I know there's a bunch of steps there that we're going to kind of go through them a little bit fast today. But if you follow those step-by-steps, you should be golden. So any questions on any of that before we start rocking and rolling? No? Cool. So before I start, got to keep the lawyers happy. The QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. So here is the agenda for today. So we just went through all of that. We'll do some of the beginners type stuff at the beginning of our time together, and then slowly but surely make our way into some of the more advanced features, and then we'll have a summary at the end. So first things first, what is Ingenuity Pathway Analysis? Well, what it is it is a software where you can take your omics data, so whether that's gene expression data, metabolomics, proteomics, microRNA data, and you can feed it into the software to use causal analytics to find key pathways that are relevant to your data set, but also IPA will generate some prediction information. So it'll let you know, hey, based on the patterns seen in your gene expression, it looks like this pathway is activated or this pathway is inhibited. Then along with that, you're able to get even more granular by looking at things like pathway maps. So over here on the left, we're looking at a chart of a bunch of canonical pathways, and those are cell signaling and metabolic pathways that are enriched in this particular data set. And then all of those colors, which we'll talk about today, are showing predictions of inhibition or activation. So drilling down into those pathways on the right here, I've taken one of the pathways and have blown it up. And here we can see all of the key players in this pathway here. So these particular molecules seem to be exhibiting some sort of effect on these downstream molecules, and it looks like this guy might be a regulator, upstream regulator of this molecule, so on and so forth. So I'll show you guys how to open up these networks and have a deeper look at your data, and then we'll also talk about how to make those networks from scratch and employ some really cool overlay tools onto those networks. Another place that we're going to be going today is I'll show you how to do a comparison analysis. So over here on the left-hand side, what I've done is I've grabbed a few core analyses that I had set up previously, and I've put them into one comparison analysis. So you can see here, we're actually looking at all of the canonical pathways that are enriched in two distinct data sets. So we have one data set right here, that's disease versus normal. The other data set over here, which is treatment versus control. Now, these two data sets are put side by side, and we are presented with all the canonical pathways in the form of a heat map here. So these colors, once again, are showing us inhibition or activation of these pathways with the blues and the oranges and the whites. And then we can take it one step further and blow that up into another heat map here, where we have taken the genes from a particular pathway. Let's say that we're grabbing the phagosome formation pathway, and we're listing out all of the genes that are contributing to this pathway and exactly what they're doing in your data set here. So the reds and the greens are the actual measurements coming out of your data set. And then once we're done with that, like I said, we'll be building some networks from scratch. So we can take one molecule. In this case, it looks like we're trying to see how APP influences the formation of amyloid beta plaques. So what we did with this particular view is I just simply dropped APP onto a blank canvas, dropped in formation of amyloid beta plaques right next to it, and then I said, hey, IPA, can you draw me any connections between these two items and any sort of intermediate molecules that might be involved in this? And then once all of those relationships have been put into place, we can then go in with a tool and say, what happens when I upregulate APP? Well, it looks like when I upregulate APP and turn this molecule red, and turns everybody else in this network orange. So that means it's predicting a more activated state of everything downstream of it, including that formation of amyloid beta plaques. So this can be really helpful for mechanism of action studies, trying to figure out what key components of your network are going to cause the biggest bang in your experiments, so on and so forth. So that's pretty much the first half of the lecture or the workshop today. I don't want to call it lecture because I definitely want you guys to interrupt me and ask questions, so I won't use that term lecture anymore. It scares me. Anyway, so the second half of the webinar workshop will be digging into some of these more advanced features. So in this particular slide, I'm showing you some of the views that we can get when we're trying to match patterns that we see in individual aspects of a core analysis. So say, for instance, my core analysis, I have the canonical pathway for TREM1 signaling that seems to be in an activated state. Where else do I see TREM1 signaling in an activated state? Out in my public data sets. So that's what this particular view is looking for. And then moving on down the road here, what if I wanted to hone in on a particular molecule? So APP, where else is this getting upregulated? Where else is this getting downregulated? And then so on and so forth with even diseases and functions. So this very last one, I'm just simply looking at activation of leukocytes and where do I see activation of leukocytes in other data sets and where do I not see activation of leukocytes in other data sets. So this is a really helpful way to chase down patterns that you see in your data to maybe bring out some interesting treatments that you might want to try, interesting new indications to explore, so on and so forth. Yeah?
Question. So this is curating the literature.
Yep.
Setting up these pathways. What about when a paper is controversial or eventually gets retracted? Does that retraction change?
Yeah, that's a great question there. So what he was asking, I don't know if you can hear that on the live stream, is what do we do? So we're going to get in a couple slides to what kind of runs the backbone of IPA and why we're able to do all this cool stuff with it. But before we get there, what was being asked is what do we do if we have, conflicting results or controversial paper that might have gotten retracted for some reason, because all of these relationships that you see here are supported by literature. Well, that is, we leave everything in. So that's up to you guys as scientists to say, you know what, I don't trust this paper, I trust this paper over this one, so I'm going to go with this result. But we will mark it in your display here. So a lot of times you'll see these yellow lines appear, and that means that it's conflicting with what APP was expected to be doing on this particular molecule. And you can just click on those lines, it will bring you it'll open up all of the literature findings that we have for that particular relationship, and you'll be presented with them so you guys can kind of figure out which ones you want to trust and which ones you don't want to trust. Does that answer your question?
So the color coding indicates what, a strength score, or?
It's the colors are going to be, again, we'll get to that in a couple more slides, but the colors all mean different things in IPA. So there's going to be red, green, orange, and blue that are going to be the main colors that we're going to be talking about today. So the red and greens are what your data set measurements are. So if something's upregulated or downregulated, you'll see it in red and green. And then for the prediction status of things, it'll be in orange or blue. So red is a more activated state, whereas the blue is going to be inhibited. But we'll get into that a little bit deeper in a couple more slides. So moving on with the comparison against publicly available data sets. So we can not only look and compare those pathways or an activity pattern of a particular pathway, but we can take your full data set and say, hey, IPA, return to me stuff that matches my full-blown analysis. So the way that we do this is we score on 4 different components of your core analysis. So we look at how matching or anti-matching your canonical pathways, your upstream regulators, your causal networks, and your disease and function downstream is. But when we get to this, we'll dig a little bit more into that, but I just wanted to introduce you to this concept that we can take your data and compare it to everything else that's out there in IPA. So one of the last features that we'll be going through today is I will show you guys a thing called Land Explorer. And Land Explorer can be really handy when you're looking at these networks and calling out specific maybe upstream regulators that look interesting, or maybe there's something that's kind of flushing out as a really cool target maybe for a new therapy or something like that. So if you wanted to take that gene and say, you know, hey, what does this look like across all normal tissue? If I upregulate this or downregulate it, is it going to, you know, kill my mice or something detrimental, right? So that's what Land Explorer allows us to do. So what we can do is click into these links and look at, for instance, we're looking at APP, so the expression of APP, and that's across all of these different normal tissues over here. So we can get a sense of how our particular target is acting across all of these different tissues. We can not only do this in normal tissue, but we have lots of links to go do this in disease versus normal tissues, treatments, oncology, non-oncology stuff. So whether your field study is melanoma or Alzheimer's or psoriasis, it's all there in these different lands. So I'll show you how to access those. Another really cool thing that we can do in Land Explorer is not only explore gene expression, but we can look at things like survival plots. So we can take a gene, so over here on the left-hand side, what I've done is I've taken that APP gene and I have asked Land Explorer, what happens if I have increased expression of APP? Does that affect the survival of my patients versus those that have decreased expression of APP? So we can very easily draw a survival plot using Land Explorer. And then we don't just have to look at gene expression. If there is DNA type information like copy number variation, somatic mutations, all of that will be available in Land Explorer as well. So here we're just looking at the percentage of altered samples that are in a particular land for APP. And you can see over here, it's in tiny font, and I apologize for that. We're looking to see what's been amplified, what's been deleted, what has multiple alterations, and what has some interesting point mutations there. So another place that we can visit Land Explorer is through the analysis match list of results. So so this view that we're looking at right here, so these are a bunch of publicly available analyses that are matching to whatever analysis it was that I called this query on. So here over on the left, we have this huge list of all of these different analyses that have been previously run. And the cool bit about Land Explorer is this link in the middle right here is going to bring you out to the volcano plots for these particular analyses. So not only will you be able to access a core analysis that has been done by our bioinformaticians by clicking on these links over here, but you'll be able to go over here and click on these links to access the volcano plots where you can go and download all of the gene expression information from those things. And I have a example of that right here. So here's a volcano plot coming from one of these publicly available studies. And then of course, if we double click on that other link on the left, it'll bring us right to that core analysis of that publicly available study. Whoops, lost my slides. Hold on one second. Okay, so now what makes IPA so special and what allows us to do some of these causal analytics? So what has happened behind the scenes is that we have a team of PhD level curation experts that comb through things like manuscripts and clinical trials and different omics data repositories. And they read all of these papers, they comb through all the supplementary tables and information there, and they boil down all of the key takeaways and findings using controlled, QC'd vocabulary. And they take that information and they compile it into our knowledge base, which powers IPA, and that's got over 2 decades of this curation, which then in turn has given us over 12.2 million research findings that are all available kind of at your fingertips. And my thing is not showing, thank you. Let me pause for 1 second here. Gets my screen duplication. There we go. Sorry about that, guys. Okay, so what I was pointing at that you guys couldn't see was this slide. Like I said, we have our team of curation experts that read all of these papers, clinical trials, so on and so forth. They boil down all the key takeaway findings into our knowledge base, and that's over 2 decades worth of information, which then gives us that 12.2 million research findings that are all available in IPA that supports all of these causal prediction that we have in the database. And you can always rest assured that what you're seeing is the most up-to-date information because we perform weekly and big quarterly updates with all of the latest and greatest research findings. So like Scott said at the very beginning of this, if you guys come across some papers or databases that you're super pumped about, let us know and we can get that into Q to get curated and added into IPA so you guys can use it in your studies there. So another thing is you don't need to take my word for it. If you just do a really quick Google Scholar search and pop in Ingenuity Pathway Analysis, you'll see here that this returned 51,000 different results here. So folks all across the globe are using IPA and trust it as a resource to help guide them in their pathway analysis endeavors across all sorts of different scientific disciplines as well. So let's start digging into things here. I'm going to get into the software in a second. However, I did want to point out that, like I said, there's a bunch of step-by-step guides at the end of this PowerPoint that you guys can use to help guide you to set things up. You can see here I have links to them. But also there are lots of other recordings that show you how to do all of these things in IPA, what it all means, how you can use it. So be sure to visit our links that we have here, or the QIAGEN website. I believe it's like bioinformatics.tv, where there are a ton of different videos out there ready for you guys to use. So one of the case studies that we're going to be using today, we're going to be looking at a paper where they were investigating the biology of anti-PD-1 immunotherapy resistance in metastatic melanoma. So here we have a bunch of patients that were treated with immune checkpoint blockade therapy, and then a portion of those patients also had some BRAF/MEK-directed targeted therapy. These samples were then submitted for RNA sequencing and profiled for clinical response and progression-free survival. And what was found in this paper is that if they saw high pretreatment levels of CD155, that correlated with high T-cell ratios of this PD-1 positive, CD8 positive to just CD8 positive T-cells, and poor response to this anti-PD-1 therapy. So that's the data that we're going to be feeding into IPA for our time today. So if you're interested, you can have a look at the paper there. So before we even start in the software, what is data analysis without some formatting, right? We need to go and play with our Excel spreadsheet to make sure that it is in proper formats to feed into IPA. So there's a couple things that you should keep in mind when we're looking at our data. So the first thing that you'll need to do anything in IPA, and this is an absolute must-have, would be a list of gene IDs. So these are the gene IDs coming out of your RNA sequencing experiments, your proteomics, your metabolomics, so on and so forth. These are going to be, an exhaustive list of, say, RNA sequencing. I say bring everything in, and then we set cutoffs in the software after we do this. So make sure you have that list of gene IDs there. And then the next things that we like to have, they're definitely, you can roll with just a gene ID list. However, if you wanted to power those, the causal analytics and the activity predictions of IPA, you're going to want to have some sort of change value measurements here. So what we have here is a data set. This is actually an ulcerative colitis data set where we have one observation or one comparison where we're just looking at ulcerative colitis versus normal samples. Then we have a second observation or a second comparison where we're looking at ulcerative colitis patients that have been treated with vedolizumab. So here we have two sets of change values that we're going to be popping into IPA. So this very first one over here, we have our full change values. So that will enable those causal analytics there. And then we have the p-value that accompanies that particular fold change. And because, like I said, we have two separate observations within this one experiment, we have two sets of fold change values and p-values here. And you can upload all of these kind of in one fell swoop into IPA, so you don't have to do individual spreadsheets for each. Now these change values can be anything from fold changes to ratios to log2 fold change, what have you, just some sort of change value. And then we accept all sorts of different file formats with text files, Excel files, and diffcuf output files as well. So with that, let's pop over into IPA itself. You guys can see that, that's great. So before we start, let's have a look at this quick start screen. So this is very handy when you're first starting out, trying to figure out how to get things done in IPA. This will give you a lot of kind of quick start guides on how to do things as well as links on how to contact our support team. So we have a technical support team that are absolutely phenomenal, and they get back to you rather quickly. Go ahead.
I have a question on the live stream. Someone named D'Angelo92 is asking, is there a way to include genes that do not have annotations in your analysis?
Yes, that's a great question. So we'll get to that when we get to the data upload. So since IPA is powered by literature findings, if there's no literature findings supporting it, therefore it won't be in IPA. So some of those more experimental findings might not be available there. So unfortunately, some of the stuff is just too new, or it hasn't been studied enough to be able to upload that into IPA. But if it is something that you guys are really jazzed about, you want to get this information in there, there's some stuff that's been published on it, certainly send us an email and we can send it over to the curation team to get that into IPA in future updates. So yeah, this quick start screen has a lot of cool stuff on it, so be sure to click around and have a look when you are in IPA. So I'm just going to close out of this to give us a little bit more space here. So what we're going to do, like I said, I am going to fly through data upload right now, but don't worry, there's other videos of how to do this, and I have step-by-step slides. So when you guys have your data, you can use that to get going with that. But what we're gonna do here is everything pretty much starts with this create new button at the very tippity top corner of the screen here. So I'm gonna give that a click, and you'll see that we have a list of all sorts of different things that you can do in IPA. So what we're going through today in IPA is just scratching the surface. There are so many other things that are available in IPA to help aid your research. So if you have any sort of research questions, experimental questions, that you're like, I wonder if IPA could do this, please reach out to us and we can help you figure out if there's a tool in here to help to get that done. But first we're gonna go to this core analysis up top. We're gonna do our data upload from that core analysis button there. So as you guessed it, we'll probably visit this upload button, and this will point out to folders that you have out on your drive here. I have some demo data, and I'm just going to grab my text file or my Excel spreadsheet that has what we're going to be importing into IPA. I'm actually going to grab this Alzheimer's disease one just to show you guys what it looks like when we have multiple observations that we're dragging in to IPA. But the rest of the time we'll be playing with that melanoma data set. So I'm just going to click on the open button here, and this will bring us to a page that looks very much like an Excel spreadsheet. So at the very top here, there's a bunch of information asking you different things. Just leave it at default. When we start messing around with some of these default parameters, it really starts limiting you on what you see on the output end of things. So if you don't want to limit your research findings, just leave everything at default. What we're going to do is we're going to spend most of our time down below here, where we're going to be telling IPA what it was on our spreadsheet that we are feeding into the software. So first things first, we need to tell IPA which columns were my gene identifiers. And if we have our gene identifiers in this very first column, IPA is smart enough to figure out what type of gene identifiers they are. So it just kind of gives you a little less work if you keep your gene identifiers in your first column. So first thing we're going to do is we're going to visit this little drop down caret button here. And instead of having it set to ignore, we're going to tell IPA these are identifiers. And you'll see down below that this switched this little drop down menu over to ensemble IDs. So this is what I'm talking about when it just auto detects what type of identifier that is. But if it didn't, or if you had your gene IDs in some other column of your spreadsheet, no big deal. You can just go in and click on the different names of all the identifiers here. So no worries there. So now that we've set our first column as our gene IDs, we need to tell IPA the rest of the stuff that we've uploaded are all experimental findings here. So for this particular spreadsheet, we have 2 observations. We have a disease versus normal state, and we have a treatment versus control state. So we have 6 columns, 3 belong to that first one, and the last 3 belong to the second one. So what we're gonna do is we're gonna go to our ignore windows kind of across the board here, and we're gonna change them over to these first 3 columns are gonna be part of observation 1. So this is my first experiment in my data sheet. And I'm just gonna go across and turn them all into observation 1. You'll see down below, these drop-down screens have changed. Sometimes they're right, sometimes they're not right. So for these first 2, it correctly figured out that we have a log ratio value in this particular column right here. We have a p-value in the second column, but in the third column, this actually is a corrected p-value. So we're gonna change this from this regular expression p-value over to the false discovery rate corrected p-value. So now we're good to go with observation number 1. So now we have to go, the last 3 are gonna be part of observation 2 since it's a different comparison here. And we're gonna set that up as these 3 columns being observation 2, and then do the same thing with these drop downs. So we have our full change right here, we have our p-value, and then we have our corrected p-value that we're gonna need to fix in this drop down here. So now that we've set observation 1 and observation 2, let's go make those names something a little bit more meaningful to us. So we're gonna go over to this edit observation names and give that a click, and that will allow us to change these names and just type in disease versus normal, and then observation 2 was treat versus untreat. And then when I click on OK, you'll see that all of those titles have now updated to be more meaningful, so you don't have to keep track of what observation 1 and observation 2 is. Now, one of the things that you may have noticed as we uploaded the gene identifier names, you might have noticed that these numbers over here, these raw data and data set summary numbers might have changed. So this kind of dovetails into that question that was just asked about experimental or new findings. So what IPA is doing is it's taking your gene IDs and it's searching the knowledge base and saying, hey, do we have information on this gene? Do we know what it is? Yes, we do, and it brings it down into our experiment. So what it's doing is it's looking over the 33,000 different rows that we have in our spreadsheet here, looking to see if we have anything in the knowledge base. I'm just pointing up to the sky because that's where the knowledge base lives. And then it brings back down, hey, we have information on 32,000 of these. So that's great. You're always gonna have some findings that might not map. Like I said, they're probably too new or too experimental, and there's just no research findings out there just yet to support them. And if you wanted to see what those were, you can just very simply click on this data set summary and show details. And that will pop out a list of what has mapped and what has remained unmapped. So you can go in and see that all of these ensemble IDs might be some new funky findings that aren't in the literature just yet. Any questions on any of that before I go forward? Again, we zipped through that really fast, so don't be afraid of it. It's super easy, and I have step-by-steps for you guys. Okay, so now that we have our spreadsheet set up, we can just click on Save. That is going to give us a pop-up warning. So this is saying, hey, you don't have any metadata attached to your experiment. Are you cool with that? In our case, sure, we're fine with that. However, when you guys start running more and more things in IPA, you're going to start amassing more and more experiments. You definitely want to start adding in metadata, because then you're able to find things and compare things a lot faster using that metadata. And if you wanted to add metadata, you can back up out of this pop-up and go over to this metadata tab right here and add in whatever metadata that you see fit. But in our case, I'm just going to click on OK. And here I'm going to choose a folder to save this in. So for those folders that you can set up in any way that you'd like to store your data, maybe by project or experiment. Here I'm just putting it into my example KO data, and I'm just going to name it test. And I'll click on save. Now what I did right there is I told IPA I'm taking this data set and I want to perform a core analysis on it. So it automatically rolls into the wizard for creating a core analysis. What it's doing is it's saying, all right, you selected this data set. It looks like this data set contains gene expression information, but if it didn't, you can always click on this and choose tox, or maybe it was a metabolomics analysis. And then it asks, what change value do you want to base your analysis on? You can choose whichever one you like, so log value, ratio, what have you. But in this case, I'm just going to leave it at default, but if you wanted to change it, just click on that little caret. I'm going to click on next now. It's going to bring us into our core analysis setup screen. So at the very top of this next screen are once again going to be a bunch of different filtering parameters that I highly suggest you guys just leave at default, because when we start clicking and applying filters, we're going to be limiting what we see on our output. So if we leave all of this stuff up top all at default, you're going to get the most kind of bang for your buck here. Unless you had a very compelling reason to limit yourself to only looking for findings that are present in mouse or something like that. But again, highly suggest look at everything and then go back and apply filters if you see fit. What we are going to do is spend some time down here in our set cutoffs area of the screen. And this is where we're going to take our 32,000 analysis ready molecules across observation, and we're going to cut down on the fat of the experiment. So when we're doing something like RNA sequencing, everything isn't important to us, right? There's a bunch of stuff that's just not differentially expressed or doesn't really matter to our experiment. So we want to filter that stuff out so it doesn't add any unnecessary noise to our experiment. So what we recommend folks to do is to take this 32,000 number or whatever number that you might have, and we want to get it to between 300, excuse my terrible chicken scratch writing here, to 2,000 to 3,000 findings. That's pretty much the sweet spot where we're zeroing in on the things that are important to the biology of your experiment and getting rid of some of the other stuff that might introduce some noise here. So the way that we're going to narrow things down is we're going to apply some light filters here. So the first thing that we want to do is apply a p-value filter. That's usually a good starting point. So here we can say, hey, I only want to focus in on things that are differentially expressed with a p-value over, we'll just say 0.05, since it's everybody's favorite p-value. And I'm going to go up here to this recalculate button, and you'll see this 32,000 number drop down. So when I do that, you'll see we have 2,900 analysis ready molecules across observations. So we're in that sweet spot. We can go forward with this, or you can go and try to drill down even further, and maybe you could apply a full change cutoff. So over here, this is maybe where you'd want to say, I only care about stuff that's being down-regulated at least one-fold or up-regulated one-fold. And then I'm going to click that recalculate button again. So that drastically narrowed things down. Go ahead.
What would you put in these cutoffs if the data has already been filtered outside of yet?
Great question. Really great question. So I highly suggest not filtering data before it goes into IPA. So take your full data set, upload it into IPA, and do the filtering within IPA. However, if that's not possible and you're just left with a filtered by p-value type data set, Feel free to pop that in here. Just make sure that you're not just uploading a list of all the upregulated stuff or a list of just the downregulated stuff, because then your data is going to be very biased. So highly suggest putting in your whole data set without any filters whenever possible, and then applying the filters in here. Great question though. So in this case, I'm just gonna clear out those full change cutoffs, hit recalculate. 2900 is a great spot to be. Now, if you remember, we had two different observations that we are uploading in this data set. So this 2900 is across observations. So we want to double check to make sure that both of those observations have enough genes to hit our sweet spot. So the way to do that is to go over here in this preview data set area right here. We are looking right now at our disease versus normal. And it looks like we have 1,500 analysis ready molecules in that one. So we're good to go on that. And if we click on the drop down, we can see we have our treated versus untreated. That one has 1,600 different analysis ready molecules. So we're good to go there. So that's just a nice double check to make sure that one of them has, you know, doesn't have like 300 and the other one has 3,000 or something like that. So now that we're good to go with that, all we're going to do is click on the Run Analysis button, and that's going to send it off to IPA space where it gets calculated and the analysis happens kind of in the cloud, so you can shut off IPA, go to Starbucks, come back, set it up at the end of your workday, and it will be ready for you to look at when you come back to it. It takes generally about 15-ish minutes, give or take, depending on how large your study is, how busy the servers are, yada, yada, yada. But usually it's right around 15 minutes, and it will send you an email when it's ready to rock for you to open it. And it will also, if you have IPA open, will flash a yellow banner at the bottom of the screen saying, hey, your analysis is ready to go. Any questions on any of that? Yeah, definitely recommend taking your full data set and bringing that into IPA. So rather than click Run Analysis, I'll hit cancel, but not before I answer a question.
So when we have the concurrency lock back in place, you know, can someone set up a run, log off, then someone else can log on?
Yep.
Set up their run, log off. They have to log off for the next person to come in?
Yeah. So when you log off, it doesn't stop your analysis. It'll still keep doing its thing in the cloud. Yep.
So far, everything you've said is this is, you know, you could load all these kind of data into one of the, you know, the online portals like Metascape or Go Analysis. And so I presume what you're going to get to is that Ingenuity Pathway is going to give you more stuff than you would get.
Yeah.
So that's like, at least from my standpoint, that's what I'd like to know.
Yeah, no, exactly. How does this differ from things like Go or GSI or something like that, right? Yeah. So that's a really good question, and we can point back to my slide that I went over a couple slides ago.
Or maybe you could just keep going, and I guess you're going to tell
and
us
highlight. Yeah, I can actually. Let's do it that way then. Yeah, definitely. So let's go back to our analysis. So if there's no other questions on this setting up, I'm just going to cancel out of this because we're not going to actually run this analysis. I pre-cooked a analysis for us to go through today, so that's why I'm canceling out of that. So to get to any analyses that you've done in the past, you're going to be doing that through this project manager window. And you'll see here, there's a ton of different folders that I have set up over my time at QIAGEN, containing all sorts of fun stuff. But for today, I have put our melanoma metastatic cancer data analysis in this example KO data folder. And we'll see here, here's our data set files. So this is the file that we've uploaded before we went and started our analysis. So if you wanted to start a fresh analysis, you can grab your data set from these data set files here. And then down here, we have our actual analyses. So these are our core analyses that we've already run. So I'm just gonna double click on the melanoma core analysis here. And I'm just gonna very quickly hop over to this pathways tab, because this is really one of the stars of the show here. So the question of what makes IPA better, I'm going to use the term better because I work for QIAGEN and I do think this is a better software, or different. Let's call it different. What makes it different than things like Go or GSI or any of that stuff? So these colors are really what's making it different. So not only are we looking for enrichment, and that's what things like Go are looking at, saying, yep, this bucket of genes that are all annotated to this one particular biological function are all present and accounted for in my gene expression experiment. What IPA does is it takes it a step further and it says, okay, not only are these genes present, but based on the expression patterns seen, so that directional data that we've uploaded into our data set here, based on that directional data, we think this pathway is inhibited, or we think this pathway is activated, based on all those target molecules and what they're doing in your data set. So hopefully that's a nice kind of clear, concise, show you why this is different than other things out there. And again, that's all thanks to that QIAGEN knowledge base, where our team of curators, they're going through all of these papers. So they're not just pouring through like abstracts and taking key takeaways from abstracts. They're reading the papers, they're looking at the supplementary data. They're taking all of the little points that were made in the paper and they're putting it into that knowledge base. So it's searching against those 12.2 million research findings to say, hey, when I see this gene expression pattern in a data set, it means that the tumor microenvironment pathway is in an activated state or it's in an inhibited state, so on and so forth. So hopefully that answered your question. Yeah.
Yeah, I've got a question. This is I want to call it drawback, or for lack of a better term, a drawback I've run into with like GSI, Geo, some other things. Mm-hmm. Let's say you have a really broad pathway, like tissue development.
Yeah.
I've had work where within the tissue development pathway, you've changed many genes.
Yep.
But let's say it's a 50-50 split. Some are up, half are down. But the shift changed the cellular identity of those treated cells.
Yep.
But most bioinformatics pathways will not call that pathway as being enriched because it's half and half.
Yeah.
But that pathway and that shift is very biologically important. Can IPA differentiate that?
Absolutely, yeah. So it will show you that these genes are present, right? So in an enriched way, and show you that, yep, your data set has all of these genes. And then it will show you either it's in an activated state or an inhibited state, but there are going to be some pathways that are just so complex and doing all sorts of different things that there's no way for IPA to say definitely this or definitely this, because maybe it's activated in this cell type versus it's inhibited in this cell type, or under stress it does this, under this it does this. In that case, these pathways, and when we go back to the canonical pathway chart, I'll show you there's gray. Those bars will be gray, and then you can click into those bars to display that network map. And you'll get a 30,000-foot view of all of the things that are happening inside your pathway, and it'll actually be distinctly, usually compartmentalized of, here's what it's doing in these types of cells, here's what it does here, here's what it does here. So yeah, great question there. Great question there. So before we start really digging into those canonical pathways and what's going on with them, let's just talk about those colors in IPA. So there's 4 colors that are very important to know about. If you're colorblind, no worries. We can actually change these colors to be more colorblind friendly. I know red and green kind of stink when you're colorblind, so we can fix those if that's something that you need to do. But the first ones that we want to call attention to are these red and green. So these are your actual data set measurements. These are the things that are coming straight up from your RNA sequencing experiment, your microarray. Anything that is green is going to be a downregulated molecule. anything that is red is going to be an increased measurement there. Now, using those actual data set measurements, that's where we start getting our activity prediction. So anytime you see an orange color, that's going to be a predicted state of activation versus a blue color, which is a predicted state of inhibition. And how IPA does that is through the use of calculating z-scores. So what's happening here is we're taking your actual gene expression data set measurements and we're comparing it to what we have in the knowledge base for a particular canonical pathway or upstream regulator, comparing it to, hey, the knowledge base is saying these genes do this if this was in an activated state. So when we're looking at our data underneath that canonical pathway chart, we're going to have a couple columns here. So we're going to have our actual data measurement here, and this is what this title means. It's your actual data measurement. And then we have this expected column. So this expected column is shorthand for expected if activated. So we're always comparing against the knowledge base, looking to see how matching is our data set to an state of activation in our knowledge base. And then we're calculating a z-score that's showing us how matching or anti-matching that is. So over here on the left, we have our actual gene expression data measurement from this pathway, pathway X we'll call it. So pathway X is enriched for all of these genes in your data set. Here's our measurements coming out of RNA sequencing. Now, when we look in the knowledge base for pathway X, we expect to see these genes upregulated, these genes downregulated. But if we compare that to our actual data set, some of that is not matching. In fact, most of it is going to be anti-matching. And you'll see here I have a bunch of little black X's and only a handful of green check marks, meaning it's matching. So since our data measurement does not match what's expected for activation, therefore it's in an inactivated state. So it's the opposite of activation here. So we're going to calculate a z-score that's going to be negative. So there is an actual formula that is a bunch of squiggly lines and scares the heck out of me because I'm math phobic, but it is available and it's in the white papers and manuals if you're interested in it. But pretty much what it's doing is it's just matching your data to what's expected for activation. So
shouldn't you, if it's activating, it would have a strong positive z-score, but wouldn't that also mean that it would have a strong negative z-score for inhibition?
So strong is kind of relative. So the strong, large numbers are just going to be dependent on how many things you're matching. So if you have a pathway that has 700 different molecules that are contributing to it, your z-score might be more along like 20s, 30s, or something like that. Whereas this, we're just looking at a few genes, so we're going to have lower z-scores, but it's primarily that positive, negative is really what I want to take home to you guys, is positive z-scores mean that your data set measurements are mostly matching what IPA is expecting for activation. See all of our little green check marks. So therefore your z-score is going to be positive. The more matching it is with the more molecules that are contributing to that z-score, the higher the z-score is going to go. So you're going to have different shades of orange. So really strong orange colors are going to be a bigger, more matching z-score than a lighter shade of orange. And you'll see that in the chart there. So before I continue on from this, how do we feel about z-score calculations, activity prediction? Does it make sense? I know this can be a kind of complex thing to wrap your brain around, so I have also included some slides in my slideshow that explain this from different vantage points, because I know people learn in different ways, so I've kind of tried to explain it in a few different ways. But it seems like we're good. If not, we can always chat after this too about it more. So we're going to go back into our software, and we'll see here that we have different shades of orange, different shades of blue. So just like we were talking about, these have a stronger matching kind of signature to what was predicted in IPA, in the knowledge base, because we have a more orange color versus this one that's a little bit more lighter orange, so on and so forth. Same thing with the blues. Another thing that you'll notice, yep.
So sorry to interrupt.
Yeah.
Our forum today is dinner theater, so we should get pause for a moment and let people grab lunch.
Yes, dinner theater. Go ahead and grab your lunches.
Folks who are remote, I know.
Yeah, make your sandwich at home.
Just a quick pause if anyone needs to use a bathroom break or anything. There's plenty of lunches, so don't be shy. Grab a lunch. Grab two if you're really hungry.
Even if you didn't sign up and send me an email, grab a lunch.
Thank you, Matt.
And I'm around for questions too. I had my belly full of Dunkins.
There's more hungry to feel free to.
I ate my body weight in Dunkins on my way in.
So essentially what you're saying is that the main difference is IPA is taking into account directionality.
Bingo. Yeah, that
As opposed to
causality.
simply enrichment of genes.
Yep. I like you using that analogy of bag of gene versus bag of gene and not even what's in your bag of genes and what it actually looks like and what it's doing. So all of those causality, all the blues and the oranges, the predictions you would get from.
And one thing that one people has done is we have 3 kinds of genetic data. We have genomics and then another kind of genetic data. And he was using IPA to merge them all, come up with looks like a very complex map.
Yep. Of all the parts. Yeah.
Just trying to. Yeah, I mean, we're both writing a paper now, and I just want to understand exactly what she's done and whether it's meaningful.
Yeah, absolutely. Some of the network stuff, the structure stuff, the complex network maps and things like that. But things like that, when you have, I just wanted to show you an example there. We can send it walk through it and talk about it. Say, that way.
I think she's had email back and forth, but I don't think she's actually had a session.
Yeah, so one-on-one. Yeah. Or even you, if you wanted to sit and go through it, one day or all of us together, I'm more than happy to do that. But still gets real complex. There's so many bells and whistles in IPA.
Is there any another database that we use the so-called L1000 database, which is a broad database? Does IPA have any connection to that?
Oh yeah, there's lots of great information in IPA as well.
And what is it?
Megan
What's
defense. Defense. We set up the whole
All of them, to be honest.
The literature base versus the L1000 data.
She just got the
Yeah,
okay.
so we have literature based and data. So things like L1000.
Like specifically, is L1000 actually, if it's actually in the IPA?
I don't know if it is, but I can check and see if it is, but I'm sure that's a big
When are you taking off?
but I can double check and see.
Because that could be useful if the IPA has L1000 plus other literature curated abilities. The idea is you have transcriptional signature of the disease state.
It should not be out.
Look for the drugs that produce the opposite.
I'll show you, so that's exactly that activity search and analysis match and pattern search. It's exactly what that is for. Take your analysis and fill it in, here's what
is normal, and that data across all of our public data stuff. And it will return to you. That's mapping. Here's how that matches. So that's not Tuesday returns. So that could be a bunch of trees. You have to know. So then you got to
Yeah, the ones that are the opposite, that are normal, those would be things other than drugs.
Could be other things, yeah. It could be a periodic rest. It could be other diseases that are still not that have this sort all of these data sets to say, I only want to look at control data sets, but then you really go down to the another cool thing when we're doing network stuff is overlay. So you build this giant network, and then you turn on this drug overlay. You can actually overlay in and say, all right, what drugs does IPA know that target all of these different things?
Yeah, see, that's something because that's a way of validating elements of the network. Put in a drug that is predicted to upregulate, downregulate the network, so that it cures whatever assay or mitigates whatever abnormal output you're getting in that system.
Yeah, so you can overlay that. You see me moving my legs on the pad, and that drug has a node. The node is just a call. When you add in that node, it's actually then in silico. You say, what happens if I drop this drug into my system? What happens to all of the targets downstream? It's a color in red saying, I'm inducing this drug. What's the pattern of effect? So that's another thing that's separated apart from
Yeah, because I guess that's a little, it sounds like it could be a little bit comprehensive. There are portals that allow you to go into the L1000 database and find a drug that is doing it for disease state. But I guess, it's basically another way that it's using our data.
It's going to be vibrating Yeah, we're good? All right, let's go.
All right, everybody's munching down. I'm going to keep going. Feel free to get up, do whatever you got to do. Munch on your chips, open your bags. It's fine. I can talk really loud. Everybody can hear me, by the way, too, right? Yeah? I feel like my voice carries pretty well, so. Okay, so we were on canonical pathways. We went over prediction information, the oranges and blues, and the reds and the greens. So now we're presented with this canonical pathway map. So here is a list of cell signaling and metabolic pathways that, number one, are enriched in our data set. And we can tell that because on this axis over here, we have our inverse log p-value. And this is the p-value of overlap of the genes annotated to this particular pathway with your data set. So how large of an overlap is that, and how likely is that to happen due to the biology of your experiment and not just random chance alone, right? So the higher that bar is, the more enriched that pathway is within your network. Then the color is showing you the activity pattern of that particular pathway. So orange is going to be a positive Z-score, activation, and then there's the shades of orange. So the deeper orange is going to be like a heavier, a stronger Z-score versus the lighter ones, so on and so forth. Then you'll see some of these gray bars. So these are those pathways that we were talking about that are super complex usually, and they are far too complex for IPA to call a clear indication of activation or inhibition just with the data that we've put in. So I'm going to click into one of these pathways to show you what that means. So that's usually the best way to investigate these pathways. So for instance, for our melanoma metastatic samples data set here, we have this antigen presentation pathway, highly enriched in your data set, but it's gray. So let's just double click on this bar. When we double click on the bar, it will display this network map. So this is a network map of all of the pieces of this antigen presentation pathway. And you'll notice that there's some colors here too. We have some faint greens, and that is showing us that we have an increased measurement in our gene. So anything that's in red and green has been actually measured in your data set. Anything in white has been measured by your data set, but did not pass cutoffs. So it's not going to be showing up as a color. And then if it was shown in gray, it just wasn't part of your data set at all. So in this case, it looks like we have a bunch of things that have been measured, but they might not have passed cutoffs. Then we have some activity prediction going on in this pathway as well. So you see some of these blue colors, interferon gamma over here, looks like it's blue, which means that we have a predicted inhibition of that particular molecule, so on and so forth. So this is like the 30,000 foot view of what's going on in the antigen presentation pathway. And we can see here, the reason why it's gray is because it's got a whole bunch of different arms that are doing different things within these cells. So we can't call a definitive activation or prediction. However, what we can do is when we open up this gray pathway and have just a general look at it, we can say, you know what, I see mostly blue kicking around on this pathway map. Everything looks like it's pretty much green that's being displayed here. So I'm just gonna call that kind of in my own brain space as a pathway that's probably tending towards an inhibited state. So that's one way to interpret those gray complex pathways. Now, these network maps, every single thing that you see on here means something. And I'm definitely not going to pretend that I even know what all of those things are, because there's a lot. So what I'm going to show you guys is go up to our help page up here. And when we click into that, you'll see that we have this legend. This legend is going to tell you everything that you'd ever want to know about these network maps. So all of those shapes that you see in those maps have a meaning behind them. For instance, kinases are triangles, microRNAs are these little trapezoidy type dudes. I don't know what that shape is. Scrolling down here, as we're hovering over some of those lines, you'll see that these acronyms start popping up. Here's the decoder for all of those acronyms. The lines with the arrows mean stuff. So feel free to bookmark this page. It's going to be your guiding light when you're going through some of these network maps. And then of course, here's our colors where we have our greens and reds and oranges and blues here. So again, that's up in help and then legend. And then we have a mini legend over here as well. And while I'm on the topic of talking about help stuff, the other really helpful thing in IPA that you'll find are these question marks and little play buttons peppered throughout the software. So if you click on those, it's going to bring you to the help page or a video description of what you're looking at. So it's tailored to the view that we're looking at right now. So if I were to click on those right now, it's going to bring us to what is a network map? What are all the things in these views, so on and so forth. So if you ever are trying to figure out what's going on, those are very helpful places to go. One thing that I do want to mention here, so as I'm hovering over some of these lines here, you'll see those acronyms pop up and then a number in parentheses next to those acronyms. I don't know if you guys can see that, TR parentheses 2. When we double click on these lines, that's going to bring up a relationship summary of how these two things are connected. So what that 2 is telling us is that we have two papers or references that are saying that it has a TR relationship. That TR acronym, we can look in the legend, or when you open it up, you'll see that it means translocation. So in those 2 papers or literature sources, we have 4 findings. So 4 distinct findings that are saying there's a translocation relationship between this complex and this complex in a little bit more detail right here. If you wanted to learn more about these complexes, you can just click on the links. It'll bring you out to a gene view page that will give you all the information that you'd ever want to know about that molecule or complex in human, mouse, and rat. Or if you wanted to see all of the findings that are powering these relationships, so again, going back to that moment where you were asking about conflicting papers, something that's been retracted, click on this view relationships between link that shows up in the relationship summary, and it will list out all of the findings and all of the papers that are supporting that particular relationship here. In this case, we have 4 findings from 2 separate papers that are supporting that relationship. Yes?
Will it list a retraction notice?
Yeah, so it will have like a little note next to it letting you know. And then also, it has that nice little quick synopsis when I go back to it. I clicked out of it a little quickly, but it literally will tell you kind of what's going on in that finding as well from this page. So those are the canonical pathways, and that's the network maps. Now back to this, this view. So here's our bars for our canonical pathways. We double clicked on this gray one to have a deeper look at it. But if you just single click on some of these bars, you'll notice that down here changes, where you'll get a list of all of the genes that are involved in the pathway that you've just single clicked on. So in this case, I just grabbed the neuroinflammation signaling pathway. So here are all the genes that are contributing to this pathway. Here's what they're doing in your data set. Here is what IPA is expecting if this pathway were to be activated. And then there's a whole bunch more cool information over here to help you with your research. Things like location, if you're looking for druggable targets, right? Maybe something on the plasma membrane would be more lucrative than some of the other spots. Have there been drugs out there that target said target? So here you can see all of that information as well. So there is a ton of stuff here, guys, to go through. Now, another place that we can stop at really quickly is our upstream analysis. So now that we've looked at all of our cell signaling and metabolic pathways that matter to our data set, what's driving those changes, right? What are the upstream regulators that are creating a change in my neuroinflammation pathway, signaling pathway here? So when we click on upstream analysis tab, that's exactly what we're looking at here. So over here on the left is a whole list of upstream regulators. The cool part about IPA, along with all of these causal reasoning that it does, is it predicts what upstream regulators might be playing a part in your data set, even if you didn't measure them. So in this case, we have a ton of upstream regulators that are showing up in our list that do not, where did my mouse go? That do not have a expression log ratio value over here. So these were measured in your data set, but these were not. And you can see that maybe they're drugs or some sort of chemical reagent or just something else. So this is another place where IPA looks at the pattern seen in your gene expression data and says, hey, this pattern looks like the same pattern that I would see if interferon alpha were playing a hand in being an upstream regulator for your data set. So based on the expression, interferon alpha might be playing a part. Looks like it might be causing an inhibition of that upstream regulator. And here's the Z-score for that particular regulator. And then over here on the right, you can see all of your target molecules that are involved in that particular regulator network. So this can be really helpful in trying to determine why these changes are happening in your data. Also in the upstream analysis tab, so not only can we look at these upstream regulators, but we can go one level above that and look at things like causal networks. So causal networks are, you have your downstream regulators. I'm gonna draw a really haphazard drawing right here, so I apologize. So here's all the genes in your data set, these little green balls. Those are being influenced by an upstream regulator right here, right? This box is our upstream regulator. But more often than not, there is a more upstream regulator that's targeting this downstream regulator, which then in turn targets your data set molecules. So it's like one more degree of separation of regulators here. So upstream regulators, 1 degree of separation from your data set molecules. Causal networks are more than 1 degree and above, so 2 and above. And when we flip over to that tab, you'll see that you'll have a whole list of new regulators to explore. Now, one thing that can catch your eye here is that this list is huge. So we have over 1,000 different causal networks to look through. That's a lot, and there's going to be a lot of stuff here that just doesn't matter to us. So what we've done in IPA is we've included a way to filter through all of this data. So every single one of these columns that you see in causal networks and upstream regulators all have the ability to be filtered. So you'll see these little filter icons show up all along the way. So this is usually how folks will narrow down to things that matter to them. So the first thing that you could do is say, I only want to look at activation Z-scores over 2. So just a little fun fact for IPA, anything with a Z-score over 2 is considered significant in IPA. So 2 is going to be the golden number for Z-scores. So anything below -2, anything above +2. So I'm going to put in 2, hit apply, and you'll notice that this 1,500 list will now get knocked down to 424. So we're getting there. We're getting to a more manageable list. Now you could go in and say, I only want to look at transcription regulators. So under molecule type, you could say, I only want to look at transcription regulators here. So I'm just going to click on that, click apply. Now we've filtered down our list to 76 things. Now, for instance, if you were trying to hone in on upstream regulators that contain very distinct genes, you have your list of favorite genes. If you want to see only the regulators that are hitting those genes, you can use the filter over here for target molecules and type in whatever your favorite gene is. And that will filter down this list now to contain only upstream regulators where that is in the network of this upstream regulator. So this is another great way to slice and dice your data as well. So while we're in this, I'm going to now introduce you into some of the network building aspects of IPA through the use of these upstream regulators. So we're going to go and grab one of these upstream regulators. I'm going to grab one with only a handful of targets in it just so we don't tax the internet system. All right, so I'm going to choose STAT2. And now what I'm going to do is I'm going to go and display this as a network. So I want to look at the network of STAT2 and how all of my targets in my data set are behaving. So I'm going to click on display as network. It's going to bring up this network map where we have STAT2 as my upstream regulator in the middle, and then all of my data set molecules kind of radiating out around it. And you'll see that we have, all of these have a color, so it's green or red because they've been measured in our data set. And then STAT2 has not been measured in our data set. It's just being predicted to be inhibited based on the pattern that we see around all of these target molecules here. So this is the first place that I'm going to introduce also some of the searching against publicly available data. So what if you were to open up this network from your data set? You see STAT2 is super important to me. All of these target molecules are doing something super interesting. Where else do I see this pattern going on out in the publicly available literature? So I keep referring to publicly available literature. And what I mean by that is when we're doing the curation of all the literature, the databases, all the stuff that's out there in the world and bringing it into IPA, we are also grabbing raw data from some of these publications, some of these databases, like Cancer Genome Atlas or Links database, and we're dragging it into IPA. We're using similar pipelines to process all RNA sequencing data in a similar fashion, all microRNA in a similar fashion. So it's a little more apples to apples when we're looking at things side by side in IPA. So that tallies up to be about 120,000 different data sets that are all reprocessed, harmonized, and ready for you guys to explore in IPA. So think about having to do that on your own, going out to PubMed, grabbing all of that data, reprocessing, reanalyzing. It takes forever. It's already been done for you, and it's all harmonized across the board so you can look at things side by side. So what we're doing here is we're taking our data set that we've run and saying, where does this pattern show up in those publicly available data sets? Now to do this, we can click on this pattern search button right here. This pattern search button is going to take all of the target molecules as well as this upstream regulator and say, where else do we see this pattern of activation? And you're going to be presented with a volcano plot that looks not super volcano-y, kind of smiley. These kind of show up a little bit funky, but anyway, the gist of this is that we have our p-value or our significance over here. So the higher it is on this axis, the more significant these findings are. And then we have a z-score. So we calculate a z-score that shows us how matching or anti-matching our pattern is to other publicly available data set patterns. So over here, we have a bunch of different analyses. So each one of these dots represents one analysis in IPA. So all of these guys over here are matching because we have a positive z-score. Anytime we have a positive z-score, matching. And then over here on the left, we have a bunch of anti-matching data sets. So over here with negative z-scores, this is where we find patterns of activation of this STAT2 network that are opposite to what we see in our data set. And then if you wanted to drill down into these actual analyses themselves, all we have to do is just draw a box around these dots, and it will bring up the information down here. And this is where we can once again use the power of filters to filter to stuff that we care about. So we were discussing over the lunch break, maybe we want to only look for data sets where a treatment has been applied. We want to find treatments that have a similar STAT2 pattern of inhibition. So over here on the right-hand side, we can just filter to comparisons that have treatment and rule out all the other stuff. So now we can kind of use a wildcard search. Did I spell that right? Yeah. So that's another tip and trick for IPA. It loves wildcard searches. So if you're going to look for something like treatment, as you saw there, I just put in the word treatment and it returned nothing back to me. But if I put an asterisk after treatment, it treats it as a wildcard search and brings back all the treatment information there. So keep in mind, use those wildcard searches when you're looking for anything in IPA to make your life a little bit easier. So are everybody good with pattern search stuff? Again, this is a really great way to look into other data sets that are showing similar biology to yours or anti-similar biology to yours. to maybe look and see, are there other indications where this could be something cool to follow up in? I have this drug that does this thing to STAT2. Maybe it'll be great to do the same thing to STAT2, but affect a different disease or function down the road. Yeah.
Is there a link there? It's a little hard for me to see, but it says GSE and then some number. Is that the link to the?
This is a link to Land Explorer.
Oh, Land Explorer.
Yeah, so we'll get to that too. Yeah, so these are really cool because actually, let's just do it right now while we're in this page. So in this pattern search page, there's a couple things that we can do here. So let's say this one is super interesting to me, this one individual analysis. I can either double click on the analysis name to look at the actual core analysis that has been pre-run for you by our curation team. And you can go and look at canonical pathways and upstream regulators, all sorts of metadata there. Or you can see here's our pathways. It looks just like where we were before. Or you can click on this comparison, this GSE kind of link over here. The header is comparison ID, but these are links out to Land Explorer. So what this is doing is it's taking and looking for that particular comparison in Land Explorer, and it's going to return back to me a volcano plot of all of the genes that have been measured in that particular experiment here. So this is the volcano plot of all of the genes that were measured for this particular study here. This GSE 67. Where'd my mouse go?
My question is that GSE number, is that the same number that you would find in the gene expression omnibus?
Yes. Yes.
Or an NCBI SRA?
Exactly. Yeah, exactly. So not only can you look at the data, but yeah, that's the accession number from NCBI Geo, from SRA, or wherever it is.
Yeah,
so that.
Like if we wanted to then go back and download it.
Yeah, and even I'll do you one better than that actually. So when we're in IPA and you're looking at things, if we look over here on in this box right here, this is all the metadata for this particular analysis that we are highlighted on here. If we scroll through this, you'll see a whole bunch of different metadata. You'll have that link out to Land Explorer right there, but you'll also see here's your sample IDs, your GSE number, and then at the bottom, here's some links out to the PubMed article for this particular study, and then also here's a link out to the data itself as well. So it's all right there for you guys to grab. So there is pattern search. That's one way to look at things and compare what you have in your data set against publicly available data sets. Now, one level below that would be something called, let's go back to our pathways. It's called activity plot. So in a case where maybe I'm interested in this PD-1, PD-L1 cancer immunotherapy pathway, maybe I want to see where else is this in an activated state, this one singular pathway. We can do that by looking at activity plot. So this button down here will go and search and say, all right, you have an activated PD-1, PD-L1 immunotherapy pathway. Where else do we see that? So by clicking on the activity plot, we'll be brought another volcano plot that looks somewhat volcano-y. This one's going to be a little bit more funky usually. It's a little more bushy. But we can see here that here are on the right side of the page, here are all of our matching. So all of these data sets have an activated immunotherapy, PD-L1 cancer immunotherapy pathway. That's a mouthful. And then over on the left, we have anti-matching data sets. So these are data sets where this particular pathway is inhibited. So these are another way to go and try to find data that matches or anti-matches to, again, look and explore other places where similar biology is happening to your particular studies. Yes?
Back to that last graphic you just showed.
Sure.
Could one say this study has some issues because there's a lot of low p-value dots?
So these are, remember, all of these dots are individual studies. So this is not just one study. This is not just one. These are matching a full analyses. So when we grab all of these, you'll see once again, here's a list of analyses.
Okay, but where would you draw the horizontal line in terms of significance?
It depends on how closely you want to match, right? So maybe you're just loosely trying to find things that match this pattern inhibition or activation. It's completely dependent on what you're looking for, kind of thing, and how wide you want to cast your net. The thing that I would mostly hang my hat on would be the z-scores. So z-scores over 2 would be super matching, super anti-matching. And then of course, if you wanted to take into account p-value, higher up is going to be more significantly matching versus lower down. But again, that's driven by your research questions. But z-score is definitely the one that I can say 2 and over. Thanks for coming, guys. So we've gone through activity plot, pattern search. Let's look at the other way to compare against publicly available data, and then we'll go back to our generating network maps. So in our core analyses, you'll see that we have this tab here called analysis match. And this is where we are going to be matching all of the patterns seen in your data to all of the patterns seen out in the publicly available data. So what I'm going to do is I'm just going to flip back to my slides real quick. I have this graphic that I wanted to show you. So all of these studies that are in IPA and all the ones that you guys are putting into IPA, when we have analysis match turned on, what it's going to do is it's going to calculate a z-score for these 4 key components of your analysis. So the first one it's looking at is it's saying, how well do your canonical pathways match the publicly available data? So a high positive z-score is going to be matching, a low negative z-score is going to be anti-matching. Then it goes and scores your causal networks, so your 2 degree and over degree of separation from your data set molecules, you know, your upstream regulators here. How well do those match the publicly available data to your data set? Again, negative z-score, anti-matching, positive z-score, matching. Upstream regulators, so your data set molecules, what upstream regulators are driving the changes seen in those molecules. It's going to give that a score as well. And then finally, it takes all of this into account and looks and says, you know, the patterns that we see in your data set genes, what type of diseases or functions do those data set molecules kind of highly align with? How enriched are those data disease enrichment diseases and functions in your data set? So it'll generate a z-score for that as well. So it takes into account those 4 z-scores, and then it does add a 5th one in where it just averages out all 4 of those z-scores. And that is what we see in analysis match. So when I flip over to analysis match, you'll see there's going to be a ton of analyses listed out here. We have 104,000 different analyses that are in analysis match. And we have our z-scores over here, and they are going to be much larger than what we were used to looking at before. So we're in the 70s and the 80s over here. And then the inverse of this, let's just quickly resort, low to high, or high to low over here. Here's our negative z-score. So we have negative 80s, 70s, 40s, so on and so forth. So all of these blue colored ones with these negative z-scores, these are analyses that are anti-matching to your particular data set. And you can kind of figure out what matters the most to you. Are you focused on matching or anti-matching for canonical pathways or for upstream regulators? Or maybe all four matter to you. In that case, maybe you want to use the overall Z-score. That's the average of the four. So it's driven by your research needs and what you think is most important for your particular experiments here. So once again, you can double click on these analysis names and they will bring you to the core analysis. Or you can click on these links over here to bring you to Land Explorer, where you can look at the gene expression information for these particular analysis. And I'm going to resort by Z-score. So again, all of these columns are filterable. So you don't have to sit and parse through these 100,000 different analyses that are present in IPA. You can maybe narrow in onto, I only care about melanoma disease. So I could say melanoma. So I'm going to use my wildcard search here and add my asterisks and then click on apply. And now it will narrow in onto only data sets that have melanoma as the disease state here. And you can scroll through all of that here. You can narrow in by comparison types, by Z-scores. So this is usually where a lot of folks just take this general Z-score and they assign an arbitrary cutoff of, say, I don't know, in this case I would say like, I don't know, anything over 60. Positive 60, negative 60. It'll give you my top hits, my bottom hits for matching and anti-matching here. Now, this is a nice place to foray into comparison analysis. So say you have a bunch of these data sets that are super interesting to you and you want to see them up alongside your analysis. Instead of looking at a bunch of individual core analyses, you can make a comparison analysis to look at all this stuff side by side. And to do this, you can either go to this create new and then go to comparison analysis, which will give you a wizard where you can go and grab data sets and drag them over into this box, this analysis to compare box, and click on view comparison, which will generate a nice little heat map type situation for you. Or you can do it from this analysis match screen or your pattern search screens. So in this case, I'm just gonna grab these top 3 data sets and say, these are super interesting to me. I wanna see them alongside my personal data set. Here I'm gonna go to this view comparison button, and that will put those alongside our data. You will get this pop-up just saying, hey, keep the p-mount values kind of in mind as you're looking across these data sets, because they're using different reference universes. Maybe one was RNA-seq, the other one was microRNA, maybe something else was phosphoproteomics, so on and so forth. So it's one of those, just kind of keep that in mind that you're looking at like apples to pears and not apples to apples here. So when I launch this comparison analysis, again, you can either do it from that create new, or you can do it just like I showed you today. Here is a different way to view core analyses side by side, where we have our 4 different data sets. So over here, we can see here's our data set. This is the first one in this row. And then we have our 3 data sets that we just grabbed from our analysis match, all side by side. And this very first screen that we're looking at, these are all of the canonical pathways. So one thing that scientists love to do first right out of the gate is change this sort method. Because right now we're just sorting on Z-score, which might not be super helpful. One of the things when you're looking at data like this, it's helpful to do hierarchical clustering. So here we have this availability in the sort method to change to hierarchical clustering. So now it clusters everything together based on things happening in the biology, right? So this will help kind of hone in on things that are important to you. You can also do that with your columns. So if you have a ton of data sets, you can do up to 20, by the way, side by side here. You can cluster your columns as well. So here you can see that these 2 data sets are similar and these 2 data sets are also similar. Now this heat map will recapitulate across all of the other different tabs. So upstream analysis, here's your upstream regulators, that same list of all of the potential upstream regulators that are in hand. what they're doing in terms of z-score, causal networks, same deal. Now in canonical pathways and these upstream analysis, we have a little bit more information we can get from these views. So within these heat maps, I'm just gonna sort this hierarchical real quick. So in these heat maps, say I'm interested in LPS, right? LPS is always gonna be at the top of the list for pretty much everything because it creates big effects, right, in any data set. So say I wanna look at this LPS regulator network a little bit deeper, and I wanna look at it in my data set. So I can click on the box right here for my data set. So this is the first data set in the column. And that'll open up the LPS network here. And this is a massive network. So there are a lot of spokes here, but you can see here's all of our data set genes that are contributing to that signal seen for LPS. So same deal like we were looking at in our individual core analyses. Now another really cool bit about this particular view is once we're selected on a data set, an upstream regulator like we are right now selected in this box, we can go to this gene heat map and create a gene heat map of all the genes that are contributing to the signal for, in this case, LPS. So I'm going to click on gene heat map and you'll get a red and green heat map that's looking at all the genes that are annotated to be part of this LPS network here and what they're doing. So once again, you can take that sort method and turn it from expression over to hierarchical clustering, where now you can cluster together things with similar biologies. So again, the grays and the whites, so the white means that it was not measured in your data set. The gray in this case says that it has been measured in your data set, but it did not meet those cutoffs that we applied in our core analyses. And then the red and greens are upregulated or downregulated. As we're going through all of these different views, every single one of these are exportable. So throughout IPA, you will see these icons of an export to Excel or an export for pictures if you want to drop them in your presentations, for lab meetings, so on and so forth. So all of that is available there. So now that we've gone through a lot of information about core analyses, let's just take these last 10 minutes and we'll go through network construction here. And I'll show you some of the cool tools that we have available to us to play around with some of these networks. So what I'm gonna do is I am going to just search for some low-hanging fruit things to get us kicked off here. So what I'm gonna do at the very top here, I'm gonna look in my search box right here. I'm gonna make sure I'm selected on this genes and chemicals, and I wanna go search for the APP gene. I know APP is important in Alzheimer's, so we're gonna try to connect that and build a network using APP and formations of amyloid beta plaques. So here we get our search results that will list out all the things that are matching APP. In this case, we just have the one. So what I'm gonna do is I'm gonna just click on my little checkbox next to APP, and I will click on this add to my pathway. This will add it onto a blank canvas that we can start building from. So here, add to my pathway. It will ask, do you want to add it to a saved my pathway? So if you guys made pathways before and you just want to keep adding to them, you can certainly do that. Or you can do this new my pathway, which is a blank canvas here. So now we see we have APP all by its lonesome in the middle of this blank pathway map. So I'm gonna go back to the very top and I'm gonna search for amyloid beta plaques. Or you could just search for, Alzheimer's, you could search for a multitude of different things. But the one thing that I definitely want to do is I want to switch my search from genes and chemicals over to diseases and functions, because that's what we're looking for now, is we're looking for amyloid beta plaque formations. So again, typing in amyloid beta, switching to diseases and functions, and I'll click on search. That will give us a whole list of all sorts of different amyloid beta plaque hits here. And as you go down the list, you can pick and choose the ones that matter to you. Another guiding light for looking through this list is over here on your right, associated molecules. The more molecules that are associated with the thing, the more umbrella-like that term is going to be. So if I want to make sure that I'm casting a wide net, I want to choose things with more associated molecules. But in our case, what I personally am going to be going after with you guys today is this formation of amyloid beta plaques. So what I'm going to do is I'm just going to click the checkbox next to that. It has 18 associated molecules, and I'm going to add to my pathway. So once again at the top here, just clicking on that add to my pathway button. It's going to ask, do you want to put it in a new one? Do you want to put it in a saved one? Do you want to add it to the pathway that we've already begun building, which is this new My Pathway 3? Click that last one. One of the questions it's going to ask you here is selecting source. Do you want to add your disease or function? Yes, absolutely. Or do you want to add your molecules or both? If we were to choose molecules or both, it's going to add all 18 associated molecules alongside that formation of amyloid beta plaques. That could be fine, but that number also tends to be somewhat on the higher side of things sometimes. So if you don't want to end up with like a big hairball of a bunch of things, just choose disease or functions. So we're going to click on disease or functions, and that should drop that disease and function onto our pathway map. And at this point now, we can draw some connections between these two items. So in our case, we want to find out how APP affects the formation of amyloid beta plaques. So what we're going to do is we're going to go over into our build toolbox right here. And this is where we can grow and generate these network maps. There's a few different ways that we can go about it. And if you wanted to find more details on all of the different functionalities, definitely visit that question mark or the video icon there, and it will go through everything for you. But for today, we want to look at distinctly how APP affects formation of amyloid beta plaques. So in this case, I'm going to use this Path Explorer tool. And here, I'm going to assign this APP gene over to my Set A box over here. So I'm going to click on my Add, and that's of course after I've highlighted APP. I'll go over and highlight formation of amyloid beta plaques, and I'm going to add it to my Set B. And then in the middle here, we'll see this directionality box. We can tell IPA in what direction we want to go here. So in our case, I want to find out how APP affects formation of amyloid beta plaques. So I want to go from Set A to Set B. Or you can go in reverse, or you can go in both ways, whichever floats your boat here. So once I've done that, I can go down to the bottom here and click on Apply. When I do, we will generate a list of the shortest paths between APP and formation of amyloid beta plaques. So there'll be, no intermediate kind of hops between these two molecules. It'll be straight from APP to formation of amyloid beta plaques. And right here, we'll see down below that we only have one shortest path available here. So what I'm going to do is I'm going to add that shortest path into our pathway by clicking on the checkbox, and then of course clicking on that Add to My Pathway. And you'll see now we have a relationship line between these two. And if you hover over it, you'll see that we have that C, which is, I think it's causation. And when we double click on the line, we'll see that we do have a whole bunch of findings, 17 of them to be pointed here, that support this causation relationship. Thanks for coming.
Thank you.
Now, if you wanted to go one step further and include relationships that might have one intermediate kind of molecule between these two, we can change this view down here to shortest paths plus 1. And that will list a whole bunch more items that we can now click and add into my pathway. And you'll see here, now we have a bunch more relationships that we can explore with some intermediate molecules in the middle here. So this is a great way to explore and build networks and find out how things are connected. Another thing that can be really cool to use in your research they're going to be available in the overlay toolbox right here. In overlay, we can use something called molecule activity predictor. And for this molecule activity predictor, this is our in silico experimentation tool. So let's say we want to see how APP affects that formation of amyloid beta plaques when it's overexpressed or downregulated. So in this case, we'll choose this red paint bucket, and that will allow us to upregulate the expression of APP. So when I click on the red paint bucket, I'll just click on APP and color it red. Now it predicts everything else except for that serpina-3 to also to have a pattern of activation here. And then the vice versa is if I go and grab the green paint bucket and downregulate APP, should turn those guys blue. So this is a good way to figure out what would happen if I were to upregulate, downregulate, or interfere with these genes in my pathway. You can do this not only from these pathways that you're building, but from your upstream analysis networks, from your canonical pathways, what have you. You can play around in there. Another really cool overlay tool that we can talk about too is drug. So say I want to find, are there any drugs out there that target any of the molecules that are in my network map right here? This is super cool when you're looking at canonical pathways, because you have a lot of targets to go through there. So in this case, we only have a handful, but let's add in a couple of these. As I'm clicking on these checkboxes, you'll see that they're being added into my network map. And if I want to find out more about them, all I have to do is just double click the drug name and it'll bring me to a chemical view report for that particular drug, which gives me all the information about it, including all the clinical trials that it's been involved in. You can also do the same thing with biomarkers in this overlay functionality. So right here, here's a whole bunch of biomarkers that have been called out in my network map right here, and I can add them into my pathway and double click on them as well to get more information. So those can all be very handy tools here. Looks like we have like 3 more minutes. While we're just wrapping things up, another place, we visited that Land Explorer through our analysis match, and we looked at volcano plots. But what if I wanted to look and see how is APP expressed across, say, normal tissues? Or I want to see how is it expressed across cancer samples. So what I'm going to do here is you can either double click on the APP molecule node in here, or you can go back up to your search by genes and chemicals. Either way will bring you to a link for APP right here. This is actually a link that you can click, and it will bring you to a gene view page for APP. Close that so it doesn't freak out. And again, here's everything that you'd ever want to know about APP. But the one place that I wanted to call your attention to is down here. So this is where all the Land Explorer data links are. So in our case, I want to look and see how is APP expressed across normal tissues. So I want to look at APP expression in the GTEx database. In this case, I'll just click on that link and it will bring me to Land Explorer, to a box plot of APP expression across all of these normal tissues. So this can be a great way to have a quick look and see what some of your targets are doing in your data sets. Now, if we go back here, maybe I want to look at APP and how it acts across cancers. So here I'm going to go over to the Oncology Consortia links and click on that TCGA. And that will now bring me to Land Explorer looking at APP across all the TCGA, Cancer Genome Atlas, samples here. So we can see how APP is expressed across all of these different cancer types. So with that, that ends all of our stuff that we're going to go through today. We went through a lot, definitely, today. So we went through how to set up a core analysis, looking at canonical pathways, upstream regulators, how you can create a comparison analysis, and then we built some networks, and I showed you some cool tools on how to test things out in silico. We also looked at how to explore publicly available data through things like activity plot, pattern search, and analysis match, all sorts of things like that. So today is certainly not the end of our relationship. My email is in on that title slide, so if you guys ever have any questions, please feel free to reach out to me. Or if you ever have any questions, this ts-bioinformatics@QIAGEN.com. This is our wonderful support team. They are a wealth of information, and they get back to you really quickly. I will also send you probably the slides that you can send out to folks, where you'll be able to see I have all these step-by-step slides on how to do data formatting, uploading into IPA, so on and so forth. So I just want to thank you guys for spending some time with us today, and I hope you think IPA is as cool as I think it is, and I hope you guys use it. Cool. Any questions?
Thank you so much.
Of course.